So the identification of clear brain behavior associations that might represent appropriate targets for the development of novel treatments is one important step on our way to precision psychiatry. And in the past, most studies pursuing this goal focused on diagnostic categories. That is, researchers used to compare individuals that met criteria for a specific psychiatric disorder to healthy volunteers. And while we've learned a lot from these studies regarding brain activation and connectivity, we also had come to realize that the diagnostic categories do not map very well onto brain neurobiology. So as a consequence, the field has moved towards alternative phenotyping strategies, such as the utilization of symptom dimensions that cut across diagnostic categories. And while these symptom dimensions are distinct, they are also moderately correlated. So one question is, how can we parse the neuromechanisms that are shared between these symptom dimensions or unique to one of these symptom dimensions? And here, latent variable approaches might come into play. Now, latent variable approaches can be divided into theory-driven approaches such as bifactor models or more exploratory techniques. And in the present study, I used one of the more exploratory latent variable approaches that is called canonical correlation analyses, or short CCA. Now, while CCA actually has been around for quite some time, it's only fairly recent that it's becoming more and more popular in the field of clinical neuroscience and psychiatry. And that's because by its design, it holds the promise of actually being able to deliver clear brain behavior associations. And why is that so? Well, what CCA does, it takes two sets of variables as input. So in our case, symptom dimensions and brain connectivity patterns. And for each of these input sets, it derives a set of latent variables, here termed U and V. The size of this set of latent variables is determined by the size of the smallest input data set, which in this scenario is likely going to be the symptom ratings. So for example, if we input 29 symptom ratings, we will get 29 latent or canonical variates. Now, what's so attractive about CCA is that the weights that we use to derive the latent variables from the input data set are derived in a way that the association between the first latent variable from one data set and the first one from the other data set is maximized. And the same is true for the association between the second pair of latent variables under the restraint that it has to be orthogonal to the first pair. And then it goes so on and so on for all the other pairs. Now, in the past, this technique has been successfully used in community dwelling views to identify well-established but relatively different domains of psychopathology. And in this study, I focus on treatment-seeking use and three common but highly correlated domains of psychopathology that are irritability, defined as an increased proneness to anger, anxiety, and disruptive behavior. So the question is, can we use canonical correlation analyses to identify shared and distinct neural features that underlie these three symptom dimensions? And a secondary question we had regarded the robustness of these findings. So would these associations replicate in an independent sample? For that reason, we did not only analyze 183 youths that were recruited at the NIMH, but in addition, we looked at a replication sample of 326 youths that came from a publicly available data set curated by the Child Mind Institute. Let's take a peek at the sample characteristics, because I think that's important. So both cohorts were comparable with regard to sex, race, medication, and parent-reported anxiety, irritability, and disruptive behavior. However, the discovery sample was slightly older had higher scores of IQ and a higher proportion of individuals that did not meet criteria for a diagnosis, as well as individuals that met criteria for disruptive motor circulation disorder. Plus, there were less cases of ADHD relatively in that sample. And use in the discovery sample reported less symptoms of anxiety and irritability compared to the replication data set. From both cohorts, we got one high-resolution T1 weighted structural image and 10 minutes of resting state fMRI data. But of course, there were differences with regard to scanner models, field strength, and other acquisition parameters between the two cohorts and also within the replication dataset, as it's a multi-site study. And here we see quality indices for the data derived from the MRIQC pipeline. And roughly summarized, one could say that it appears the quality of the structural image is higher on the replication dataset, whereas the quality of the resting state functional data is higher for the discovery dataset. Now, both data, or like imaging data from both cohorts were submitted to the fMRI prep pipeline. And we all know that resting state data is very susceptible to motion, and we controlled for motion. However, we did not do scrubbing. Instead, we decided to regress out the motion components derived from ICA aroma along with other variables. And that's because evidence suggests that ICA aroma performs equally well to scrubbing. Plus, it has the huge advantage that it leaves the time course intact and allows us to have the exact same time points per individual. To extract the time courses, we decided for a recently published parcellation scheme with 200 cortical pulses that we augmented by 8 subcortical regions per hemisphere. And then the time courses were extracted from native space. From that, we calculated the connectivity matrices, and we decided for partial correlations, as they offer a direct as opposed to an indirect or shared estimate of the association between two nodes. Before we actually came to the part where we did the canonical correlation analyses, we also removed nuisance variables from both symptom ratings and brain connectivity matrices. And importantly for that, we used the Hujun method that does not introduce artificial associations between the two input data sets. Now, given that we had brain connectivity matrices and the high dimensionality that goes along with this, it was necessary to include a step of dimensionality reduction. And for this step, we decided for principal component analyses. Canonical correlation finally was calculated in MATLAB, and the significance of the variables were determined by 10,000 permutations. And last but not least, we calculated the cross correlations. So we applied the weights, the canonical weights from one data set to the other data set, and then determined the association to get at the replicability of the finding. Now, what did we find? In the discovery data set, we identified 4 canonical variates, 2 of which appear to be shared across input domains or cross-domain variates, whereas 2 others were specific, one specific to disruptive behavior and the other one specific to symptoms of anxiety. Well, how did we come to this conclusion? Well, we looked at the canonical loadings that are essentially the associations between the latent variable and the input data. Now, don't get overwhelmed, we work through this slowly. So if we look at this black ring, this one represents the 4th canonical variate. And as you can see, this variate was highly associated with symptoms of anxiety. So we see that 8 of 10 surpassed the threshold of 0.2, whereas symptoms from other dimensions only loaded weakly or negatively on this variable. And a similar pattern is observable if we look at the orange ring that represents the 2nd canonical variate. Here again, we see high loadings for 5 out of 7 items coming from the measure of disruptive behavior. Now, the third variable, the green ring, reminds of a previously reported shared factor of negative affectivity that is characterized by excessive and high arousal responses to negative affect. So we see very high loadings for items measuring temper outbursts, separation anxiety, panic, and impulsivity. Whereas the first canonical variant that also start to be shared between diagnostic categories shows the highest loadings for items such as can't sit still and can't concentrate, and might hence represent some sort of inhibitory control deficit. Now, which neural circuits were related to these latent phenotypes? And maybe shockingly, maybe sadly, there was not a specific neural circuit which mapped onto these latent phenotypes. More so, we saw from the loadings many, however, relatively weak interhemispheric connections, as well as connections spanning distinct networks within each hemisphere. And this overall pattern was observable for all 4 canonical variants. The only variant that stuck out to some extent was the second one associated with disruptive behavior, because the connectivity pattern was denser here and there were also more negative loadings. Now, how did the picture look like in the replication data set? In the replication dataset, only 2 canonical variants surpassed the threshold for statistical significance. And if we take a look at the loadings, we see that both of them represented shared or transdiagnostic factors. So we see high loadings in the domain of irritability, but also anxiety and disruptive behavior for the second one, and then high loadings for irritability in the first, and then high negative loadings for disruptive behavior and anxiety. With regard to the imaging data, relatively to the discovery data set, the patterns were more sparse. However, overall, again, we see many relatively big interhemispheric connections, as well as connections spanning networks within each hemisphere. Now, how did we do in terms of replicability? If we look at the set of clinical latent variables, there was one association that survived control for multiple testing, and that was association between the first transdiagnostic factor from the discovery dataset and the second transdiagnostic factor from the replication dataset. The loadings from the imaging data throughout were only very weakly correlated. So to summarize the study, our goal was to identify associations between clinical symptoms or symptom measures and resting state connectivity. And in our discovery data set, we were able to identify 4 of such latent clinical variables, 2 of them being transdiagnostic and 2 specific to symptom dimensions of anxiety and disruptive behavior. All these associations survived correction for multiple testing. And I did not show this, but particularly the transdiagnostic factors in the discovery data set manifested consistently when we systematically varied the input to participant ratio, indicating some robustness against overfitting. And it was also one of the transdiagnostic factors that further replicated in an independent sample. With regard to the takeaway, I think the most important finding from this study concerns the imaging data. So the low imaging data, we observed many weak connections between hemispheres and within between networks within hemispheres. And these connections, albeit they were stable within the discovery data set, only showed weak associations with the loadings in the other data set. So we kind of observed the absence of strongly replicable patterns associated with questionnaire-based clinical symptom dimensions. So we therefore conclude that instead of a consistent universal pattern of brain connectivity, there might be many different broadly distributed brain configurations that lead to similar clinical expressions. And from a clinical perspective, this seems relevant because it speaks to the necessity of a diverse set of approaches to identify targets for novel interventions. So last but not least, I want to thank Dr. Alan Liebenluft, who's a wonderful mentor. And I also want to thank Anderson Winkler and Dr. Daniel Pine, who were crucial in the realization of this project. Thank you.